is good afternoon, everyone. First and foremost, I want to thank Dr. Nigute madam and Dr. Avari sir for giving me this honor to speak in this prestigious platform. Uh, so, I will start with my topic. Um, so, you have learned about all basics of innate and adaptive immune system. What is immunological tolerance? What all are basics, and what are all functions of different immune cells? Now, something different from that is about senescence and what is immunosenescence and what are the molecular mechanisms and what is its importance. So why do you need to know molecular mechanisms in different diseases and cancer? So that will be highlighted in your afternoon session on immunotherapy, which is based targeted therapy for which you should know all molecular events that are involved not only in infectious and autoimmune diseases but also in cancer. So first, the brief outline of my lecture to introduce what is immunosenescence, what are the changes in different immune cell subsets that occur with this process, what are hallmarks of immunosenescence, and what all changes occur in innate as well as adaptive immune system during aging and during infection. What molecular mechanisms they play major role here, different diseases that are associated with it, and what is the key role of immunosenescence in cancer. So moving forward to introduction. So though these terms are sounding similar, there is difference between what is senescence and what is immunosenescence. So to define senescence, it is with aging, it is a normal physiological process in which each and every organ of human body, it starts slowly uh, it, uh, its functions to decrease with age. So this is senescence, but only when these changes they concern our immune system and the different immune cells to decline their function and start functioning in inappropriate or excessive manner, that is termed as immunosenescence. Okay, so we can say it is a age-related immune dysfunction that results in increased incidence of not only infectious diseases but also in neoplastic diseases, and it forms the underlying base for high failure rate of immunotherapeutic drugs in cancer patients. Also, it accounts for increase in recurrences even after immunotherapeutic treatment in cancer patients. So, what are senescent cells? What are their functions? So, these are non-functioning cells. These are the cells which have stopped dividing. Okay, they have arrested proliferation, but still they are metabolically active. And this metabolic activity of senescent cells, it accounts for dysfunction of the immune system. So, these are the cells which are characterized by permanent cell cycle arrest with accompanying morphological abnormalities, with loss of their proliferation and resistance to cell death. Okay, so these are cells which will accumulate in the tissue and resist cell death, which is one of the harmful thing. So to prevent this physiological response, these cells they prevent genomic instability, which is induced by persistent antigens and accumulation of the damaged DNA. But however, when there is excess senescence of these cells, particularly of T lymphocytes, which are the important immune functioning cells, it ultimately lead to decline in the immune function and contributing to increased risk of age-related diseases. So if there are young immune cells still persisting, they can clear these senescent cells that uh, arise with aging and disease. But with advanced age, these senescent cells, they have one property of developing pro-inflammatory cytokines and certain chemokines which are termed as SASP, that is senescence associated secretory phenotype. Okay, so these form the basic pathogenesis for inflammaging and further immunosenescence related diseases. So inflammaging is nothing but a systemic state of low grade chronic inflammation which accounts for number of chronic diseases and even low-grade malignancies that gradually start worsening with aging. Okay. So, thus we can say senescence, it acts as a robust tumor suppressor with this senescence associated secretory phenotype, which act as chemoattractant and therefore stimulate immune cell mediated clearance of these senescent cells. But what happens to this normal physiological process with aging and in cancer? These senescent cells, they are unable to get removed by inefficient functioning immune cells, and therefore they start accumulating in the tissues and resist cell death. That leads to a state of inflammation and further disease. Now, what different changes they are encountered in the immune cells with senescence of the immune system? Okay, so now you have studied what are all basic functions of all immune cells, including T cell, B cell, NK cell, dendritic cells, and macrophages. Now, what happens pathological here in all these immune cells? So, thymus, as we know, is the major lymphoid organ for synthesis of all types of immune cells. With aging, there starts gradual thymic involution. Thymus starts degenerating, which accounts for decrease in the T cell output. Not only T cell, but also B cells. So there is decrease in the naive T and B cells with corresponding increase in the memory T lymphocytes. Also, there is accompanying decrease in the T cell receptor diversity, which will suppress the adaptive immune responses. Okay. Now, uh, natural killer cells, which form important part of the innate immune system and also promoting anti-tumor immune response. They have certain activation receptors, that is NKP30 and NKP46. So, with aging, these activation receptors, they show reduced expression on the surface of NK cells. Okay, and they are responsible not only for recognition of the tumor cells but also their lysis. So, if there is reduced expression of these receptors, the tumor cell lysis will go unchecked. This is commonly encountered in hematological malignancies, skin cancers like melanoma, and ovarian cancer. Okay. Then dendritic cells and macrophages, which the major function of these cells is phagocytosis, endocytosis, and antigen presentation. So these functions, they get suppressed with advancing age. Now, what are these MDSCs? These are myeloid-derived suppressor cells. Okay, these are immunosuppressive cells which get accumulated in the blood, bone marrow, and spleen, particularly in leukemic patients. Okay, so these immunosuppressive cells, when they start accumulating in marrow and spleen, they will result in inefficient tumor cell clearance, resulting in further progression of the cancers. Now, what is the basic concept of immunosenescence? Okay, so as I have told you, with aging, there occurs gradual thymic involution resulting in the decreased T cell output as well as reduction in the T cell receptor diversity. In addition to this, the senescent cells, they start acquiring pro-inflammatory phenotype. That is, they start secreting certain cytokines, chemokines that are SASP, which again contributes to senescence of immune cells. Now, one of the characteristic property of the T cell with aging is telomere shortening, which is due to increased replication of the T lymphocytes due to decrease in the telomerase activity. There is altered differentiation and proliferation of T and B lymphocytes. Now, also with aging, we know there is disorganization of the extracellular matrix components, reduction in the adhesion molecules, which normally keeps the epithelial and stromal cells of any tissue glued and adhered together. But this gluing and adherence is lost with aging, as the stromal cells and adhesion molecules, they are lost. So that results ultimately in tissue disorganization. So all these factors, they account for impaired immune response of all the immune cells involved in innate as well as adaptive immunity, contributing to immunosenescence. And what is the ultimate effect? This will result in increased incidence as the newly encountered antigens will go unrecognized by the immune cells. This will contribute to increased incidence of infections. Also, chronically, if it progresses, in autoimmune diseases, and and also in tumor progression and metastasis. So this was the basic about immunosenescence. So this I have already talked. This is nothing different. The hallmarks of immunosenescence is the first and step to initiate is thymic involution. The rest all starts with this basic step. Then there is loss of T cell receptor diversity, which results in loss of adaptive immune response, loss of ability to respond to newly encountered antigens, inflammation, which all contribute to immunosenescence. Now, these are a few of the examples to understand how these hallmarks of immunosenescence they result in the disease or tumor progression. So, beta galactosidase it is one of the enzyme which starts accumulating in the lysosomes of senescent cells, contributing to cell cycle arrest. Now, interleukin 7 production, which is important for development and survival of the T lymphocytes, as it is decreased with thymic involution, the production of the T cell decreases. Now, these are few of the immune checkpoint related molecules which are present on the surface of NK cells, that is TIM3, CTLA4, which are the major immune checkpoint molecules. So as with aging, there is increase in the expression of these molecules. So the tumor cells, they go unrecognized or there is excessive or inappropriate response of the immune cells to tumor cells due to increased expression of these immune checkpoint molecules. Now, as we know, with aging, there is also decrease in the cytotoxic activity of CD8 T lymphocytes and the associated cytotoxic molecules, that is interferon gamma, granzyme and perforin. Okay, so this results in inefficient clearance of the pathogens as well as tumor cells. Okay, now CD27 and CD28, which are the different co-stimulatory molecules expressed again on the T lymphocytes, as their expression, co-expression is reduced, this results in corresponding increase of the immunosenescent markers, that is P16 and P21, which are cyclin dependent kinase inhibitors. Okay. Now, in addition to immune dysfunction, senescent cells, as they are metabolically active, they also result in certain metabolic derangements too, with increase in the glycolysis, increased production of reactive oxygen species, uh, which creates oxidative stress and damage to the cells. There is decrease in the mitochondrial synthesis too. Okay. Now, different examples how the innate immune system dysfunction occurs with age, with aging, during infection. So, interferon type 1, which is the major inflammatory mediator produced um, and determines susceptibility to viral infections. But what happens with aging? There is decrease in this interferon production. So, there is increase in the viral replication with early spread of infection. Now, some of the examples, best examples of this are influenza and SARS-CoV-2 infection, where there is increase in the neutrophil production that results in the lung damage. So these neutrophils normally they will get phagocytosed with macrophages, but as there is decrease in interferon and suppressed neutrophil recruitment, as well as the aged macrophages which are unable to phagocytose these neutrophils, they start increased damage of the lung parenchyma. Now this I have already speak about how the myeloid-derived suppressor cells are responsible for hematological malignancies. Now what are the adaptive immune dysfunctions with age during infection in B and as well as in the cytotoxic T lymphocytes? So as there is decrease in the naive B cells and increase in the memory cells, which contributes to inappropriate antibody production and response to vaccination. This is due to increase in interleukin 6 production by the senescent cells and helper follicular T cells, which are required or which are the stimulus for adequate antibody generation. But when they start producing increase in interleukin 10, their response of antibody production is reduced and also the response to vaccination is reduced. Now, cytotoxicity of CD8 T lymphocytes is also decreased with aging. The best example to narrate about this is elderly with COVID-19 infection. Okay, so why there is inefficient immune response in elderly to COVID-19? This is due to decreased cytotoxic activity of the CD8 T lymphocytes. Now, less studied and understood is about the effector or memory T cells. How do they function in adaptive immune dysfunctions with age and during infection? So, as there is decrease in the clearance of pathogens and tumor cells, these are the permanent residents of the uh, tissues which start accumulating after the um, response to primary antigen. But when they encounter some new antigen, they are, with aging, they are unable to respond to newly encountered antigen, and even if it is cleared, what mechanism makes it pathogenic is the persistent inflammation and fibrosis that they leave behind. Now little bit difficult or tricky to understand are the molecular mechanisms that are seen with immunosenescence, and why these molecular mechanisms are important to understand, as they form the basis for targeted immunotherapy in cancer patients. Okay, so what are these molecular targets? For that you should know first the molecular pathway. Okay, first is when there is persistent antigenic stimulation of uh, already damaged T lymphocyte, which is having depressed immune functions, or the stress induced damage to the T lymphocytes, okay, which makes it molecularly inactive. Now, this is due to certain genetic and epigenetic changes, such as abnormal DNA methylation patterns, modification in the histones, and certain non-coding RNAs, which function here. So what is this persistent antigenic stimulation? With chronic antigenic stress, it induces an inflammatory state resulting in activation of macrophages. But when there is such repeated stimulation, this results in poor response to the newly encountered antigens. And what does it suggest in elderly? That there is occurring shift in the immune system towards immunosenescence when it is not responding to persistent antigenic stimulus. Now with aging, we know there is also increased production of reactive oxygen species. And what is the role of these reactive oxygen species? They induce oxidative stress in a cell, making it cellular, uh, leading to cellular senescence and decline in its function. It also induces apoptosis and autophagy in oxidatively damaged cells. Now, what are the genetic and epigenetic changes? Several transcription factors, okay, which are required for DNA methylation, such as MYC, and related to T lymphocyte proliferation, activation, and differentiation. With aging, there is decrease in the expression or decreased levels of these transcription factors. Now, the best examples of such abnormal DNA methylation patterns which are encountered in elderly patients are cerebrovascular and cardiovascular diseases, where the abnormal DNA methylation is noted in the blood and endothelial cells, leading to atherosclerotic progression and MI. Okay. So this is the basic mechanism why there is increased incidence of MI with increasing age. Then in osteoarthritis and osteoporosis, there are changes in DNA methylation patterns of cartilage and bone. Now, how microRNAs they contribute to immunosenescence? They mediate chromatin remodeling due to damaged DNA, release a number of inflammatory mediators which interfere with the development and differentiation of T and B lymphocytes, decreasing their immune responses. Okay. So the molecular mechanism to understand these genetic and epigenetic modification that occur in the T cell pool. Now in the previous lectures you have already learned how are the T cells they functioning in the removal of encountered antigen or the pathogen. Okay, so the naive T cells which are dependent upon oxidative phosphorylation to meet their energy demands, on getting activated when they encounter the antigens, they activate mTOR signaling pathway, okay, which starts secreting cytokines such as HIF-1 alpha, increasing glycolytic metabolism as well as mitochondrial mass, and this results in epigenetic remodeling of the effector T lymphocytes. Simultaneously, some intermediate metabolites they also start increase in the nucleotide synthesis. Okay. So this is followed by antigen clearance, but whatever T lymphocytes which are left behind as memory T lymphocytes, they start showing a metabolic switch with decrease in the metabolic activity of the T lymphocytes. So again they use oxidative phosphorylation to meet their energy demands. Now this is the normal T cell pool functioning. What happens when there is persistent pathological stimulation of the T lymphocytes, either by repeated encounter of the antigens, by repeated exposure to the tumor cells, or due to increase in the T cell receptor diversity. When this happens, the T cells, they get exhausted and they are unable to function properly. So they start generating abnormal signals that result in inappropriate metabolic activity of the cell, with increase in the aerobic glycolysis, decrease in their cytotoxic activity, and decrease in the mitochondrial synthesis. Now finally, with aging and stress, what happens? All types of T cells, they turn out into aged and senescent cells which are having inefficient immune functions as well as decrease in the metabolic functions. So these cells are inefficiently cleared by the defective immune cells with advancing age, and this is the mechanism how they start accumulating and resulting in the disease process and worsening. Now the different diseases which are associated or which get worsened in elderly due to dysfunction of the immune system. First are cardiovascular diseases, which we have already learned. Then autoimmune diseases. Now the major mechanism for encountering increased incidence of autoimmune diseases such as rheumatoid arthritis with elderly is premature immunosenescence, for which the basic mechanism is again the increase in the telomeric attrition, that is telomere shortening due to decrease in telomerase activity, and the pro-inflammatory phenotype which starts secreting pro-inflammatory cytokines, and they form the basic pathogenesis for disease severity in rheumatoid arthritis. Now the best example for neurodegenerative diseases which are encountered in elderly is Alzheimer's disease. Again the mechanism remains same here. Now why this is one of the recent advanced topic for research, how immunosenescence it plays a key role in tumor progression and worsening. It is due to epigenetic modifications, then mitochondrial dysfunctions, and the cellular senescence, which accounts for cancer or the more aggressive tumor phenotypes that are encountered with advancing age. Also, the tumor response and tumor aggressiveness differs between young and older patients. Now, what is mechanism for this? Effect of aging on TME, that is tumor microenvironment. How the tumor microenvironment differs in young and in elderly. Okay, so this is important to understand. Why immunosenescence is important, or which contributes to worsening of the disease in elderly. As we can see, in the young tumor microenvironment, there is much of the integrity not only of the surface epithelium but also of the extracellular matrix, which is more organized, and with age there starts disorganization or loosening of surface epithelial cells as well as disorganized stromal cells. So such disorganization of the stroma and surface epithelium, it facilitates easy spread of the tumor cells from surface epithelium to stroma, then metastasizing to blood. Okay, so this loosening of the extracellular matrix as well as of the surface epithelium is seen with advancing age. There is loss of integrity, okay, so which facilitates tumor progression and spread. Now, in addition to this, the aged stromal cells, they or the senescent cells, they also start secreting pro-inflammatory cytokines. Increase in the pro-inflammatory cytokines such as interleukin 6, 8 and 10, which are major contributors to inflammaging. Okay. Now again, in addition to this, there is accumulation of certain immunosuppressive cells such as myeloid derived suppressor cells and regulatory T cell, which again accounts for tumor progression and metastasis. There is senescence of T and B lymphocytes, then dendritic cells and macrophages uh, with advancing age. Okay, now, so this is how all these factors they contribute to early tumor progression and metastasis in elderly individuals, contributed with the senescent cells and the inefficiently functioning immune cells. Now, it is the T cells which are important, T cell pool which we have learned previously, that result in anti-tumor response. So, how is this tumor microenvironment contributing to tumor progression in cancer patients? Whatever adenosine monophosphate that is produced by the tumor cells, okay, it is being transferred to the T lymphocytes, which start activating this PKA-CREB pathway, okay, which is damaging to DNA. Also, there is competition for glucose by the tumor cells and regulatory T cells, which such glucose deprived state, it again leads to ATM related DNA damage, activating these downward pathways and ultimately causing increased expression of cyclin dependent kinase inhibitors, that is P16 and P21, which are important for cell cycle regulation. So this results in cell cycle arrest. Now, as we have learned with senescent cell, there is also increase in the T cell receptor diversity. So their down regulation, it again activates P38 pathway, causing down regulation of TERT and again resulting in DNA damage. In addition, this T receptor diversity, it activates mTOR signaling pathway, which starts inactivating autophagy and increased production of reactive oxygen species, ultimately resulting in senescence of the T cells. Similarly, with increased oxidative stress by the tumor cells, the damaged DNA start producing increase in the SASP. Okay. Now, after going through all these molecular mechanisms, I would integrate all this to understand how immunosenescence is important, playing a key role in cancer, and is important to be understood to prevent tumor progression and metastasis. Okay. So this is a circuit between cellular senescence and inflammation that promotes tumorigenesis. Okay, so as there is increased accumulation of the with senescent cells with aging in different tissues, this leads to a state of inflammaging, secreting pro-inflammatory cytokines, chemokines, different extracellular matrix components and matrix metalloproteinases. This results in counteracting immunosuppression of the regulatory T cells. These immunosuppressed regulatory T cell, they start generating inhibitory cytokines such as interleukin 10 and TGF beta. These inhibitory cytokines, they inactivate cytotoxic CD8 T lymphocytes, which therefore become inefficient to clear the pathogens and tumor cells. In addition to these, the NK natural killer cells, they express certain activation receptors. So there is sustained activation of these NKG2D receptors on the surface of NK cells, which gets inhibited with the immunosenescent cells, again leading to unrecognized or unimmune checked senescent cells, which start again accumulating within the tissues. So this is the circuit for different diseases which are related to immunosenescence. So if any questions.